Yeah, perfect. So, once again, like I said, warmly welcome all of us to the AfriScience webinar series with Dr. Watzani. And here, along with me, distinguished members of the AfriScience Network. Here we are. The AfriScience Network is a platform which is very popular all across Africa, promoting and supporting scientific research, innovation, and collaboration, and connecting researchers, innovators, where we share knowledge, resources, and opportunities. And without much ado, we've taken quite some time. So today is one of our monthly webinar series, and we have with us here Professor Bello. So continue with the subsequent procedures of webinar series. We're inviting Shitu. Uh, well, yeah. So Dr. Aminu, please, sir. The floor
Okay,
is
thank
yours.
you. Yeah, thank you very much, Dr. Watzani. I hope I'm clear to everyone. Can you hear me?
Yes, sir.
Okay, thank you very much,
Yes, sir.
everyone, and thanks. Thank you, and thank you so much for coming to the 16th webinar of the AfriScience Network. I would like to thank you also for allowing me to read the brief citation of the speaker for today, who is no one but Dr. Muhammad Bashir Bello. Dr. Muhammad Bashir Bello is an associate professor, associate research scientist and the head of research and development unit in the vaccine development division of the infectious disease research department at King Abdullah International Medical Research Center, KMRC, in Riyadh, Saudi Arabia. He obtained his PhD from the prestigious University Putra, Malaysia, where he developed a reverse genetics-based vaccine against Newcastle disease virus. With a specialized focus on molecular virology, comparative genomics, and vaccinology, Dr. Bello's research centers on the development of next-generation vaccines to combat emerging viral pathogens such as dengue virus, MERS-CoV, Lassa fever, and COVID-19. Dr. Bello has successfully secured numerous research grants in both Nigeria and Saudi Arabia, along with substantial international funding for a variety of vaccine-related projects. He has authored numerous papers published in high-impact scientific journals, contributing significantly to the field of infectious diseases. His research work has been presented at several national and international academic conferences. Notably, he was part of the expert team that developed the MSc Vaccinology program at the Regional Center of Excellence for Vaccines, Immunizations, and Health Supply Chain Management at the University of Rwanda in Kigali, a program designed to train Africa's future leaders in vaccine development and public health. Prior to his appointment at KMRC, Dr. Bello was a senior lecturer in the Faculty of Veterinary Medicine and the deputy director at the Center for Advanced Medical Research and Training, CAMRED, at Usman Ofodo University, Sokoto, in Nigeria. Here at CAMRED, he led several groundbreaking projects, including the development of vaccines for Lassa fever and COVID-19, making significant strides toward improving public health outcomes in West Africa. His contributions also extended to national public health efforts, where he played a key role in the Nigeria COVID-19 Research Coalition and served on the Nigeria Governors Forum COVID-19 Technical Advisory Committee. In addition to his extensive research contributions, Dr. Bello has mentored numerous MSc and PhD students and taught courses in virology, immunology, and molecular biology at both undergraduate and postgraduate levels. His dedication to education and capacity building is mirrored in his active involvement in professional societies as the International Veterinary Vaccinology Network, World Society for Virology, African Society for Virology, Biotechnology Society of Nigeria, and the Nigeria Society for Immunology, among others. His current work, which includes genomic surveillance and the development of mRNA vaccines, demonstrates his commitment to advancing global vaccine efforts and addressing pressing public health challenges through cutting-edge research. Thank you very much. And with this, I will invite our distinguished Professor Oyewale Tomori to chair this important webinar. Thank you very much, and you're highly welcome, sir. Thank you.
Thank you, Aminu, and welcome to all of you. My friend, Mohammed Bashir Bello, we're really quite excited to have you here today. And we are looking forward to what you bring in. You're raising a very important point about the need for local vaccine research and development and manufacturing in Africa. Many of you will remember a few days ago that Bill Gates came to Nigeria and said local vaccine production was not, should not be a priority. I'm preparing a paper to counter that. I should be publishing that in another few days. And I'm getting Nigerian Academy of Science to come up and say, look, Bill Gates is talking, sorry to use the word, bullshit. You cannot, in one hand, support Rwanda and Senegal to be producing vaccine. And then you say it shouldn't be a priority for Nigeria. And the excuse you gave is economy of scale. What economy of scale are we talking about? What's the population of Rwanda? What's the population of Nigeria? So this is the time we must be ready to stand up against all these do-gooders. I hope they are hearing me. But anyway, in another few days, I should be writing an article about this, that Bill Gates, yes, he's providing us with a lot of things, but he should mind his business concerning the local vaccine production. We are going on ahead, whatever it takes to do, because that's the direction for us to go. They cannot be playing double game with us. They are telling us, supporting a smaller country to produce vaccine, and telling us, Nigeria, that has the largest market, and say it should not be a priority. My brother, Mohammed Bashir Bello, the floor is yours. So I'm starting on this so that we can know what direction we are going. And we need all the support of all of you. Come out and let's speak boldly about what is happening at different countries. Bashir, the floor is yours. Thank you very much.
Thank you so very much, Prof. I hope I'm audible.
Very clear.
Thank you so very much. I want to begin by appreciating the organizers of this important occasion, members of the AfriScience Network. The network I consider the largest collection of intellectual biomedical scientists on the continent of Africa. I am really, really proud to be a part of this network. I am particularly delighted because I am invited to be a guest speaker for the 17th, if I'm not mistaken, 17th monthly seminar series, constantly organized by this vibrant network. What is even more important here is the fact that we have in our midst experts, particularly the chairman of this occasion, who I consider my great-grandfather academically, Professor Oyewale Tomori, and someone with expertise in virology, a professor of virology, and someone that has his name written with golden pen in terms of public health and epidemic response at national, regional, and global level for the past 50 years, if I'm not mistaken. So the fact that we have him in our midst is enough to add color and flavor to the entire presentation. Thank you so much, sir, for accepting to chair this very important activity. Now, without much waste of time, I would want to step down to the nitty-gritties of business of today. I'm here to make a presentation on monkeypox resurgence as a critical reminder for Africa to strengthen local vaccine research, development, and manufacturing. Now, Africa, unfortunately, has already been black painted as an infectious continent, largely because every damn deadly infectious disease, whether it is HIV, dengue, whether it is chikungunya, whether it is all these arboviral infections, talking about Lassa, also Ebola, Marburg, and so on, tuberculosis, all these are prevalent on the continent, and some of them, they even have their origins somehow traceable to the continent of Africa. Now, there are so many factors that are responsible for the continuous emergence and re-emergence of some of these diseases in Africa. First of all, in Africa, we know we have a kind of favorable climatic conditions that somehow support the proliferation and emergence of new pathogens. But way beyond that, also in Africa, we have, unfortunately, very poor sanitary conditions, which is also key in emergence of new diseases. We also know that we are characterized by lots of massive urban migration. We have very fragile health system. We are involved in deforestation. We are involved in consumption of bushmeat. We are involved in bush burning. We have natural disasters like flood, and so many factors that synergistically act together to alter vector biology and distribution, causing pathogen adaptation, and of course, emergence of new diseases. And that is why Africa, in most cases, is often seen as the breeding center of emerging infectious diseases. Whether this is entirely true or it's also another way of demoralizing us as Africans, that is a topic of discussion for another day. But the fact remains that for a disease to remain, to make global importance, it has to cross the boundaries of Africa. We have seen this with many diseases. Lassa fever, for instance, that emerged somewhere in 1968, remained neglected tropical disease for more than 50 years. Not until cases began to be exported outside the continent, then only the world felt there was a need to declare state of emergency for this disease. And now subsequently, Lassa fever was included among the list of priority pathogens for which urgent vaccine development was needed. We have also seen this with Marburg, with Ebola, and with so many diseases. Because they were found in Africa, the world does not really, pay much attention to it. This is exactly what is happening to monkeypox. Monkeypox, actually, the term itself is a misnomer, because some people are saying it is even supposed to be called rodent pox, because the virus actually exists naturally in rodents, but somehow monkeys were accidental hosts. This disease was first discovered in monkeys in 1958, and these monkeys were brought from Singapore to Copenhagen, Denmark, and they were supposed to be used for research purposes. But then the virus was detected there, and subsequently in 1960s, the virus was also detected in non-human primates in other countries, not in Africa. But because the first detection of human infection or zoonotic transmission of this virus happened in a 9-month-old baby in the Democratic Republic of Congo, somewhere in 1970, monkeypox is often seen as more of an African thing. 10 years later, we know that this disease was also restricted only within the African sub-region, so that Democratic Republic of Congo, Nigeria, and a few more countries were reporting cases. Although the cases were quite few, I'm going to discuss the factors that were responsible for the few cases in those days. But the world tend to ignore everything. By 1990s, we have seen a kind of rise in number of cases in DRC, and up to the year 2000, the world did not really consider monkeypox to be a problem, despite the fact that there was exponential rise in the number of cases in Africa. But because it is Africa, nobody cares. And of course, unfortunately, we also don't care, because this is our thing. Was to provide African solutions to African problems. We fold our arms waiting for someone to come and point out our problems. Now, monkeypox continued because monkeypox was found everywhere. Up to the year 2000, like I said, it was restricted to Africa, and so it didn't attract any global attention. In fact, editor-in-chief of Journal of Infectious Diseases, who is also an immunologist at Harvard University, he was reported to have said, over the past 30 years, Africa has reported dozens of monkeypox outbreaks, but an even untold number of cases. But the US and and other parts of the world, they really don't give a damn. They care not, because they felt it is a phenomenon not in my backyard. It is somebody else's problem. Little did they know that infectious diseases do not have respect for international boundaries. They can always fly and move, especially with the advancement of transportation system and so many other global warming factors, to mention but a few. Now, this is exactly what happened, because monkeypox did not start garnering global attention until it was exported to the United States of America in the year 2003. And this was through some small mammals that were imported from Ghana that got, contact with pets and subsequently infected a small baby, and then the cases kept on increasing. And this US CDC confirmed about 71 confirmed cases in the outbreak in the United States in the year 2003. And this is when media was bombarded with lots of information about monkeypox. Monkeypox is there. Meanwhile, it has been in Africa for the past 30 years, but people did not really pay much attention to it. One thing very common about this outbreak in the United States was there was no human-to-human transmission. But then, and again, they banned importation of these rodents from Africa, and then they were able to quickly control this outbreak. Meanwhile, continuously, the virus continued to cause little outbreaks in different countries, particularly in Africa, Democratic Republic of Congo, and a few other African countries that are considered to be endemic for the disease. This is what kept on occurring until the year 2017. In the year 2017, there was a kind of large outbreak of monkeypox in Nigeria, and this was said to be the spark of the global epidemic we are experiencing right now. Because in 2017, in May precisely, a 35-year-old man presented with some severe lesions, pox-like lesions all over his body, including his genitalia. In fact, according to the report, that the lesions were so severe in his genitals that his penile organ was about to even cut off because of the severity and depth of the ulcerative lesion that was seen as a result of this. Now, all tests, chicken pox, syphilis, and all these differentials were ruled out. At that time, because monkeypox was not among the priority pathogens that we routinely investigated, nobody really cared to check it, and that we couldn't establish exactly what was the case at that time. Now, while this patient was on admission, another patient that matched the same clinical case was also presented to the same hospital. But this one, he reported to be HIV positive, but his HIV status could not actually explain the severity of the lesions. So they documented, but luckily they were discharged. But a few months later, again, there were other ones that had advanced HIV, which happened to also get this pox-like lesions. That was when they knew that there was maybe something was going on, because possibly this could be an outbreak. Now, in neighboring Bayelsa State, there was also similar thing. Now there was influx of people coming with pox-like lesions, especially along their genital organs. And again, because of the severity and because of the numerous cases that were being seen, samples were taken and subsequently examined, and it was confirmed to be monkeypox. The first report after nearly 40 years in Nigeria. Because of this report, Nigerian government, and this is where I salute NCDC and the other relevant public health agencies in Nigeria, they swift into action, and then they initiated active surveillance for this disease, including looking for the virus in some animal reservoirs, because at that point, it was even very difficult to tell what was the source of this virus. Now, genomic studies, of course, were they were undergone. Many people, they went into these genomic studies, and it was found that the virus actually came as a result of zoonotic spillover infection. And the Nigerian outbreak, despite all these early signs, it did not ring a bell. And international agencies or international organizations did not really learn anything from the Nigerian outbreak. Now, if you look at the picture of the 2017-2018 Mpox outbreak in Nigeria, you find out that most of the cases were concentrated in the southern part of the country, with a few, of course, in the north, in Abuja, in Nasarawa and Plateau State. But this is not to say that they were not found in Sokoto, in Borno, in other northern parts of the country. Maybe this could be reporting bias or something. But then this is where I also want to say that we really need to intensify on disease surveillance system, which I will also talk about it later. If you look at the map of the scenario, the spatiotemporal distribution of this outbreak, you find out that in October 2017, that was when we saw the peak number of cases, both suspected and confirmed cases. But then it kept reducing somehow, so that by October 2018, there was no any reported case of or suspected or confirmed case of Mpox. So this is where I said we give kudos to public health agencies in Nigeria for their swift action. Now, meanwhile, the outbreak continued. Sporadic cases were seen here, there, and everywhere. But in May 2022, the first case of Mpox was reported in the United Kingdom, and this case had linked epidemiologically with travels to Nigeria, and this led to rapid expansion in number of countries that were affected, so that in no time about 116 countries with nearly 100,000 infections were documented worldwide. And one thing was unique about this 2022 global epidemic: cases have no direct link. In some of them they don't have no direct link with endemic areas. We have seen that also outside Africa, most of the infections were driven by men who have sex with men. And the virus, isolated from 2022 outbreak, it appears to mutate so rapidly. Some are saying that it mutates like 10 times faster than the standard virus mutation rate. And it was responsible, multiple viral strains were responsible for this outbreak. This is to show the importance of genomic surveillance, because how were we able to even decipher all this? It was through genomic surveillance. Genomic surveillance was what identified the root cause of the global spread, and even deciphered some of the clades and the strains and the variants of this virus that were circulating all over the world and their epidemiological linkages. I will talk about this briefly later. Now, WHO responded to the Mpox outbreak by first of all declaring the Mpox as a public health emergency of international concern. And they also launched international efforts to track cases and monitor transmission trends globally. And then they provided guidance on the use of vaccines, which I will also talk about shortly. There is also increased dissemination of public health information in order to prevent the transmission of the disease. And of course, there was, and because of all these efforts, the emergency status.
Please mute. Hazan Jankuzo, please mute. Hazan, please mute your phone.
Can I continue?
Please go ahead.
Okay, so because of this, we were able to, the emergency status was lifted in 2022, but then it was reinstated because of resurgence in 2024, precisely in August. What is this virus called Mpox? Now, this Mpox virus is actually a brick-shaped virus which has double-stranded DNA genome, and this double-stranded DNA genome is about 197 kb in size. It's pretty much a big virus, and again, you find out that the genome is organized in such a way that it has two terminal regions, what we call inverted terminal repeats, that is separated by a central genomic region, which encodes about 190 open reading frames that are responsible for production of all the proteins that are involved in evasion of immune response and those that are also inducing neutralizing immune response in the affected host. Again, you will find out that this virus, like I said, is an autopox virus that belongs to the family Poxviridae. This virus is like a cousin brother or cousin sister, whether it is sister or brother, I'm not sure, but whether it is like the cousin of vaccinia and variola viruses. Okay, so this viruses, those that were, the variola virus, we know historically has caused lots of havoc. It caused lots of pandemic in previously and led to the death of many people. I may touch about it also briefly. These are some of the the strands or clades of the virus. When you check phylogenetically, you will see that the virus has two major clades, clade 1 and clade 2. Clade 1 and clade 2, they have this geographic specificity. You will see that clade 1, in most cases, it is found in the Central African region, and it used to be called Congo Basin virus. And this virus, it is mostly found in DRC, in Cameroon, South Sudan, Gabon, and Central African Republic. It is responsible for mostly in sporadic cases, but because it is endemic, but it causes some few outbreaks. And transmission is largely through zoonotic spillover events, but then it is said to be this most severe because it has case fatality rate of about 10%. Clade 2, on the other hand, is more of West African strain found in Nigeria, Liberia, Sierra Leone, Ivory Coast, and maybe to some extent Cameroon. And then this virus has very little case fatality rate of about 1%. Now, this is the general picture that has been painted until 2024, that clade 1 is more severe, clade 2 may not really be severe. But some question was asked. According to an article published like 1 week ago, they were asking this question: is there really a more dangerous Mpox virus clade? Because if you look at this map, you will see that, you know, as the claim that clade 1 is more dangerous than clade 2 is as a result of the fact that the data that was used to make this conclusion was quite limited at that time. And if you take a look at the demography, you find out that clade 1 that affects Central African Republic, in most cases, it was affecting young children. While clade 2, that was more of an West African kind of the virus, it affected a young adolescent with mean age of around 29 years. So age itself is a factor that may play a role in determining the pathogenicity. And that is why, as the number of cases now increase, the blue line here is showing the number of cases. As the number of cases became more available, now the cumulative number of Mpox cases, as they become more available, you will see that the case fatality rate was dropping. So that even in Congo itself, the clade 1 virus, at a point as of August 2024, the case fatality rate has dropped to around 3 or thereabout. So this, we still ask this question: is there really a more dangerous Mpox virus clade, or maybe we are just making this assertion from the little data available when we made all these estimations? And that is why it was concluded that for us to say that we are dealing with a more dangerous clade, we need more clinical and epidemiological data to support all this. Now, what are the vaccines that are available for prevention of Mpox? Before we do this, then I think I like telling stories about this gentleman, Edward Jenner. He was an English physician. He was credited for developing the first successful vaccine against smallpox in the year 1976. And what did he do? When smallpox was really a problem, at that point, the only way that people were doing to really to control smallpox was through a practice of what they call variolation. Variolation was involved taking like live or legions from diseased individual to inoculate it into another healthy, apparently healthy individual, with the view to exposing them so that he develops mild symptoms and then later recover. But he now observed that at that point, you know, all milkmaids that were involved in milking, they never came down with smallpox. So because of this observation, he now he wanted to do the experiment. He got an 8-year-old boy and inoculated him with cowpox material, and then later exposed him with smallpox, and he was able to demonstrate that the boy did not come down with the disease. This was the genesis. And because cowpox, he thought, was caused by variole vaccini, meaning smallpox of the cow, so this was how the vaccine even came into being. And because of his effort, WHO launched an international global smallpox eradication campaign in the year 1959 using his own principles. And to God be the glory, by the year 1980, smallpox was declared completely eradicated. So some of the vaccines, I mean, based on his principles that are available for smallpox, were this genius vaccine. Now, this vaccine, you know, it's otherwise known as Invamune. It is a third generation vaccine. Its single dose is enough to, you know, provide substantial immunity. It is usually from a strain known as modified Vaccinia Ankara. This strain is known to be highly attenuated, and so the vaccine is also safe and non-replicating, and it becomes the vaccine, WHO's vaccine of choice for the current outbreak. This is originally a smallpox vaccine, but I will show you later why it is now being used also against monkeypox. Now, there's also another vaccine, AKM 2000. This is a general second generation vaccine. It is, however, live attenuated vaccine. People are still frowning at using this vaccine, but then it has been given approval to be used during monkeypox outbreak. There's also LC16M8, which is also approved in Japan to be used, and now it's been, and there's evidence published in the New England Journal of Medicine that neutralizing antibody response has been demonstrated in healthy adults. These vaccines are all available, and they are being used in different countries to protect against this. Why are they being used? Because of this enormous antigenic similarity. Remember I said that the Mpox virus and the vaccinia virus and variola virus, they are all cousins. Because of this similarity, they share so many proteins together. They share envelope proteins, they share vaccinia-related antigens, they share many core proteins. And some of this, the similarity is so much that it is up to 98 or nearly 99% in some cases. In fact, most the major vaccines, the major proteins, that are responsible for inducing neutralizing immune response, these ones that are available, do you see that they share up to 93, 94, 98% amino acid similarity? So that explains why it is said that up to 85%, the protection is observed when you use smallpox vaccines against monkeypox. What are the drivers of Mpox re-emergence? One of the drivers of re-emergence of this is spillover events. We see multiple spillover events between animals to human beings, and these animals could be the rodents, could be the monkeys. So because there is possibly increased contact between human beings and then these animals, then there is every chances that this infection will occur. And subsequently also you will see that because of this spillover event, there is sustained human-to-human transmission, which has been established in this outbreak. Then you will ask this question: why is it that in those days there was no sustained human-to-human transmission? Because it was said that the virus was more circulated in rural areas in those days. Somebody is saying something? Okay, so can I continue?
Yes, please. Bala, please mute your phone.
So the virus is circulating, because the virus was circulating in rural areas, and so there was the kind of thing we are observing now, that the virus is more of an urban thing than that explains why the transmissibility could be higher. So this is one of the reasons why we see re-emergence of monkeypox in this days. Another major factor that is responsible for this is waning of smallpox immunity. Now, smallpox, because of the campaign in 70s, 60s, between 1950s to 1980s, to eradicate smallpox, many elderly individuals have been vaccinated against smallpox, and so they were immune against smallpox. So immunity from smallpox has really conferred them immunity against other related orthopox viruses, including Mpox. And this may explain why the cases in those days in 70s remained quite low, because many people in those days, they already had immunity to smallpox. But as the generation, as the younger generation comes up, people that have no immunity to smallpox, there is a kind of decline in herd immunity to monkeypox, especially in younger generation, and who were born usually after the eradication of smallpox. And so the population now continuously become vulnerable, and that is why recently it was said that in Democratic Republic of Congo, we have seen up to 20-fold increase in the prevalence of Mpox compared to 30 years ago when it was first observed. And because of the cessation, therefore, of smallpox vaccination, there is a kind of gap in immunity that contributes to re-emergence of monkeypox. Another possible factor that could explain the re-emergence is, of course, the evolution of the virus itself. This is what we have seen recently. Between September 2018 to January 2022, a lot of mutations, up to 40 or thereabout mutations, have been demonstrated in the viruses that that were kind of re-emerging nowadays. And we have also observed a kind of shift from zoonotic transmission to sustained human-to-human transmission. It is possible some of these mutations are attributed to, it is possible these events are attributed to the mutations of that, but this, however, require additional studies. So what are the current challenges right now? We are quite lucky because monkeypox, by the time it came, there was already some vaccines. But the vaccine, and these vaccines are known to be efficacious. Like I said, up to 85% protection have been demonstrated. So, but the question is, there is limited global stockpiles of these smallpox vaccines. Most countries, they have stopped producing and stockpiling smallpox vaccines after the eradication, and that is why it is there is reduced availability. And countries like United States, they maintain emergency stockpiles of smallpox vaccines for bioterrorism preparedness. And WHO, which oversees global stockpiles, facilitates vaccine distribution during the outbreaks. And then because of this vaccine availability issues, you know, wealthier countries, they tend to have more of the vaccines, leaving the low-income countries, which some people are really frowning at. Are we really low-income countries? Or maybe it's something else. We're just trying to hide under that cover. So we have a kind of inadequate access to these vaccines. Wealthier countries, they tend to have more of these vaccines. And this is a big lesson for us to have our own local vaccine production supply. Secondly, if you look at it, even we have insufficient access to the vaccine, particularly Africa generally. Recently, WHO announced plans to share some 31 million smallpox vaccines in its stockpile. However, African countries may not immediately access them due to the reasons I highlighted before. And I said that Nigeria also has recently got 10,000 doses of Mpox vaccines, but you and I know that what will 10,000 doses do in a population of like 200 million people? We also know that the Democratic Republic of Congo, for instance, it was said that they needed about 300 million, about 3 million doses of vaccines to end this outbreak, but they were able to get 250,000 doses alone. These doses will go nowhere. So far, African countries have accessed less than 10% of the vaccines needed to end Mpox outbreak. And we are the most hit because we are considered to have the endemic, we are considered endemic regions, yet we don't have this, simply because we don't have facility for our own local vaccine production and supply. Even though EU, they promised to deliver 5,000 doses of the 500,000 doses of the vaccine, but when these vaccines are arriving, only God knows. Now, despite all this, to showcase the current landscape of vaccine manufacturing capacity in Africa, you find out that if you look at what I showed on the left-hand side, you see that America, with a population of 1 billion, they have capacity to produce 5.56 billion vaccines. And you see Asia Pacific, with a population of nearly 5 million, they have capacity to produce over 16 billion doses of vaccine. But Africa, that have population of 1.41 billion, they have, you know, they can only produce currently about 0.03 billion doses of vaccines. Look at the map of Africa. These are the only countries currently, I think, that have vaccine production capacity, and most of this, they are fill and finish. You know, we don't have the ones that are able to do the drug substance manufacturing. Most of them, they will just do the formulation, you know, and this will depend on technology transfer and so many other hurdles. Now, again, you know, this is about manufacturing, but if you look at also, we are talking about research itself. Vaccine research begins with surveillance. We need to really empower ourselves in terms of disease surveillance. And nowadays, disease surveillance is usually what they call genomic surveillance. And genomic surveillance in the entire Africa, look at the capacity. See countries that are able, that have a few genomic surveillance capacity based on NGS. If you check our data, in most cases in the gene bank, you find out that the sequences available for common viruses, you will find out that it is using Sanger sequencing technology, which usually provides very little information. But with NGS, you can have the whole genome, and that provides genomic information, which is very useful in tracking and tracing the epidemiology and transmission dynamic of viruses. And you will see that only a few countries, they have this capacity. And even in Nigeria and maybe other few countries, you find out that it's select places. Well, thank God in Nigeria, there's like SGID and a few other centers that are able to really involve what do you call massive training capacity in this genomic surveillance, but these are grossly inadequate. These are grossly inadequate. And in the entire Africa, you will see that 75% of this NGS capacity is usually restricted to 5 countries: South Africa, Nigeria, Kenya, Morocco, and Egypt. Now, all other countries, they will have 1 or 2. 3 countries with empty NGS capacity. So we have a very long way to go as far as genomic surveillance is concerned. And this is the foundation of our own in vaccine research. This time around, not even manufacturing. Because if we are saying we must produce vaccines on our own, we have to do vaccine research. And if we have to do vaccine research, we must intensify our surveillance. And this surveillance should be based on NGS technology. Now, we also have the infrastructural deficit also for doing vaccine research. If you look at most of our facilities, very few laboratories are well equipped. You will find out that our research equipment, in some cases, they are obsolete. You know, we find out that we have very few, state-of-the-art laboratories. And also the laboratories like BSL-3 and even 4, BSL-3 laboratories that are able to handle most of these dangerous pathogens available or prevalent in Nigeria, you find out that few countries have these facilities. So we have also a long way to go here. We need to really empower ourselves in this aspect. There's also the issue of limited expertise of vaccine research. You find out that trained vaccinologists are not quite many on the continent. And there is also insufficient specialized training. You will find out that there's limited opportunities for African researchers to engage in international collaborations, and so on and so forth. So problems are here and there. You also have problems on regulatory hurdles. Thank God now in the continent, we are now really pushing forward. I will talk about it later. But then there are lots of hurdles, complex approval processes, lack of harmonized regulatory framework, limited capacity for regulatory bodies. Like I said, many regulatory authorities in Africa, they lack the resources and expertise to conduct rigorous vaccine evaluations efficiently, which contribute to delays in approvals and so on and so forth. So what's the way forward and what are the positive vibes? Now, in spite of the earlier picture I have painted, we can still see light at the end of the tunnel. But for us to see that light, we must always remember what we faced during COVID-19 pandemic. During COVID-19 pandemic, Africa was taken unawares because wealthier nations, like I said, they secured the majority of early vaccine doses, leaving Africa with limited access. And in fact, up to mid of the year 2021, only a small percentage of Africans have been vaccinated. And the vaccine hoarding was also seen, and it wasn't with disparity and logistic issues are there, and Africa therefore had minimal local vaccine production capacity, which is what really sent us into all these problems. Now, what should we do? First of all, we must expand our vaccine production capacity. I'm glad that some few things are happening. Like, for instance, Gavi recently launched African Vaccine Manufacturing Accelerator, and then they intend to fund it with a huge amount of money, which I just captured there. And also in Rwanda, we can see that BioNTech opens mRNA vaccine production capacity. We shall discuss this much later, but then this is good news, at least at continental level. Something is happening. Okay, again, in terms of boosting surveillance, genomic surveillance strategies in Africa, genomic surveillance is something that has to do with, that has so many values. First of all, it ensures early detection of variants of viruses, which informs public health responses. And it also helps in tracking the transmission dynamics of the virus. It ultimately helps in vaccine development. And the process is simple: outbreak investigation, sampling, and then you sequence it using NGS. And then you do comparative genomics and phylogenetic analysis based on available data, which calls for the need of bioinformatics expertise on the continent. So boosting genomic surveillance strategies is key to achieving effective vaccine research in the continent of Africa. We also must build multi-pathogen laboratories. Gone are the days when we shall consider only one pathogen and say this is a lab for a single pathogen. We have to find a way of building specialized laboratories, if possible, regionally located laboratories in each region, so that we can maximize the available capacity for human resources. We can also reduce cost of equipment maintenance, and we can also do bulk procurement and then standardize and streamline workflows across the different pathogens. We also must intensify specialized trainings in vaccinology. I can see that Africa CDC is doing quite a lot. They are sponsoring people for specialized training in vaccine manufacturing, in vaccine technology transfer. And then there is this annual program that is going on in South Africa, the AfroAdvac, Advanced Vaccinology Program. These are all short-term courses that are able to train, young researchers in the field of vaccinology. We have also seen that, like I said, in Rwanda, they have recently approved, and they want to start, I think, next month, they will start MSc Vaccinology, just to produce more people that will be in the vaccine industry in order to meet up the 2040 agenda of the Africa CDC, that Africa will produce 60% of its own vaccines. During COVID, I remember we are saying that less than 1% of vaccines used in Africa are produced in the continent of Africa. So in summary, what are the lessons that we learned from monkeypox outbreak? Monkeypox outbreak circulated several years before the first case was identified. This strengthens the role of genomic surveillance for us to really be proactive. Secondly, there's a need for accessible and rapid diagnostic tools. And this is where I actually salute also our scientists that did pretty much well in handling Ebola outbreak. When Ebola came in, because of the fact that they were able to swiftly respond, it was quickly diagnosed, and then there was massive contact tracing activity, and then the outbreak was subsided. The outbreak quickly subsided. So we have to give kudos to them for this. But how did they do this? They were lucky that they have good, laboratories, especially at the borders in Lagos and other parts of the countries. But this must be decentralized. We must have more laboratories in different regions, in different countries, okay, so that we would be able to immediately contain emerging infectious diseases. Now, we have also learned that in this outbreak, it is usually, mediated by men who have sex with men. And this was shown based on certain clinical, epidemiological, and genomic observations, further strengthening the need for robust genomic surveillance apparatus. There's also the need for us to have our own local vaccine production system, because we have seen how we have suffered. We received just 10,000 doses of the vaccine. Meanwhile, we require God knows how much, but then we receive only 10,000 vaccine doses to take care of it. Now, in conclusion, I want to say that as Africans, we have really done good job in handling so many things on our own, but we still need to do more, because the ongoing monkeypox outbreaks and other neglected tropical diseases only highlight the critical importance of strengthening our local vaccine research, development, and manufacturing capacity in Africa. I remember from a few months ago, I was in Europe. I was attending a program. Somebody, he mistakenly mentioned this, not knowing after he mentioned it, he saw me, and then he felt somehow that he mentioned mistakenly that Africans, he mentioned, and I quote, Africans are like our guinea pigs. We test what we produce in them. And then immediately he said this, he saw me, and then his reaction completely changed. So we must have our own local vaccine research, development, and manufacturing. I'm emphasizing on the research and development itself, not only manufacturing, because until we can have our own product, drug substance, have our own antigen production system, we are still not, free from the bondage of those people. Secondly, Once we address challenges such as lack of advanced laboratories, shortage of trained experts, insufficient funding, and those regulatory hurdles, now we are sure that we will boost Africa's capacity to respond effectively to current and future health crises. And now the need also to emphasize on genomic surveillance, vaccine manufacturing, and public health infrastructure as a way of reducing dependence and also promoting self-sufficiency, especially during outbreaks. I remember also we do quite a lot of copy in Africa, because if we copy, sometimes we don't even copy very well. I remember during COVID-19 pandemic, when the first time I was actually involved in COVID response then in Nigeria. So what we were asked to do 2 negative testings before an individual is discharged and be free of the virus. And this was just based on evidence brought in from another country. They said that after 11 days of infection, they find out that they don't see the virus, even if it turns out to be PCR positive. Instead of us to conduct as a country, and I mean, even Africa as a whole, to conduct simple experiment and see if it works for us, we just immediately adopted it and just said, suddenly we just sent in, order that subsequently just one test is needed. So what I'm saying is, always we have to be creative, and let us also do it on our own. Test it on our own people, check it on our, things to make sure that it really works for us, and we are not just adopting what other people have worked on. And finally, I want to say that by investing in local capacity, Africa can lead the charge in tackling endemic diseases and even contribute to global health security. Thank you so very much, and this is over.
Bashir, thank you very, very much. It's been a really wonderful presentation, and you said addressed so many things. So I'm not going to go into a summary, but I'll just make a few comments and then ask people who have comments to make. The first thing I want to talk about is the fact that you raised the fact that when COVID came, we were ignorant. It wasn't only Africa that were ignorant, the entire world was ignorant. And you completed by saying the fact that we're waiting on the world. Whatever it is they did, then we use that to solve our problem. Because we are all ignorant, rather than doing what was happening in our own area, we are waiting for WHO and others to determine for us what our situation is. And it all failed, because what was happening in Europe was not happening with us. That's number one. Number two, I think all of us here sitting down here must begin to see what names that we're giving. The names that the so-called, like your man who said Africans are guinea pig over there, what names are we giving? They tell us we are resource limited. Are we really resource limited? Or we're resource squandering? Are we really resource limited, or we're resource wasting? If you don't know exactly what your problem is, you have found the wrong solution to it. So when you tell you are resource limited, you go into the despondency of self-pity. And then you say, I'm resource limited, but we're not. But when you know you are resource wasting, then you address the wasting rather than being a listing. The other issue you make, again, during the COVID, I'm bringing back all these points for each of us to begin to think about the situation. We accuse the European people of hoarding, of whatever, and that, they didn't do anything about our problem, 50 years of this. What were you doing about your own problem yourself? You have a problem in your country, 50 years, you keep going, they didn't come. When it got to Europe, they did something. You remember the fake was discovered, was stopped. The fake about monkeypox was done not because of Africa. It was done because, the situation was happening in Europe. And they stopped when the situation in Europe stopped. Not Africa never stopped monkeypox since 2017. But fake was never declared because of us. It was declared because it was in Europe. I think these are issues that you and I must begin to think about. And then when you turn around and you accuse them of hoarding and all that, I want just to reverse the situation. Suppose it was Africa where everybody was dying, that they were dying in Europe. Suppose it was Africa that discovered the vaccine. Suppose it was Africa that put its money out and bought all those vaccines. And then Europe now comes to you and says, come and give me vaccine when your own people are there. Will you give them? So we need to begin to think along this line rather than begin to have a self-pity and accusing the people of the world of having the problem and then do that. Bashir, you raised a very important point. 2014 Ebola in Nigeria was done a combination of surveillance, contact tracing, the isolation of cases, lab, something of that nature. India, I mean, the other countries did that. Kenya did that against anthrax. The same thing with Uganda against Ebola. We have the strength and the talent on this. The reason why we're not making full use of all, we all know surveillance is the pre-thing. That's the first thing you need to do, not the vaccine. Because if you survey and you limit your this, you don't need a vaccine anymore. Or what you need will be a little amount of money. So I'm going to open the floor to others to talk about this thing. But I wanted us to bring out all these things that rather than turn around the world and blaming everybody in the world for a problem, we not only we have our resources, we have the human and the talent, but if we don't put our money where our money is, where our mouth is, then of course we'll continue to be dependent upon the world. So Bashir, thank you for opening up our eyes, for opening up all the new areas we are talking about. It means it requires for us to go back to solve our own problem ourselves. That also I was glad that when African Center for Disease Control declared the emergency before the WHO, time has come for us to wake up and sort our own problem rather than wait for external people to sort it out. Finally, your slides, I hope you'll be able to show it. Well, a few people complain that they didn't see the slides. I hope the organizers will ensure that this is shared amongst all the people. Thank you very much. And then if you have questions, raise up your hand. I don't know whether Aminu has one or two things to say before we move on. Aminu?
Yes, Prof. Sir. I don't have anything to say, but for the people asking for the slides, I'm composing a message to advise them that they should go to the YouTube channel of the AfriScience Network, like that channel so that they can get updates on all the new uploads and they can watch all the past webinars.
Okay. Thank you very much.
I'm going to provide the link to the YouTube channel. Thank you, sir.
Thank you very much. Thank you. I appreciate that. Now, I've seen two hands that are raised. I hope Bashir is noting all the comments that are coming from different places. You know, about this. First of all, Mustafa Imam, I see your hand raised.
Yes, thank you very much.
And please go straight and ask the question. We don't have too much time.
Okay, no problem, sir. Okay, thank you very much for this opportunity to talk, and thank you, Bashir, for the very clear and comprehensive talk on Mpox vaccine. I was a bit worried earlier in your presentation when you talked about the fact that Mpox was circulating in Nigeria and in Africa for quite some time. And there was a comment you made that the West ignored this until it became a problem. So I was a bit worried what your implications were in this particular context, especially considering what Prof. Tomori mentioned regarding the fact that we shouldn't be blaming other people for our problems. So now my question is, I mean, who are you insinuating should be responsible for a problem that actually is happening in Nigeria, although there's a potential for that problem to expand to other parts of the world? Again, I am happy. That is the first question. Again, I am happy you talked about the issue of intensifying surveillance. And we know that the level of surveillance in Nigeria is not adequate. So what do you think what do you think immediately needs to be done, especially considering the fact that there are labs that are left dormant, which could actually all over the country, which could actually contribute to surveillance in Nigeria. And then finally, my question is, now that you're in Saudi, what do you see in Saudi and in other places that you've been to? What do you see them implementing that is different from ours, that is helping them, that we could actually, some of the lessons that we could learn, implement immediately in Nigeria to actually solve the issues of lack of reliable data coming from genomic surveillance, and also help us towards developing local vaccines for not just Mpox, but for other infectious diseases that are affecting us locally in Nigeria and in Africa? Thank you very much.
Bashir, would you like to take more questions, or you answer this one and then before calling next one? What do you prefer?
No problem, sir. Anyhow, sir.
Okay, let me call one or two more people, and then please make your questions direct to the point because of there's so many things coming up. Abidemi or Monisi, you are next. And if you have talked, please lower your hand so that we don't have to keep calling you back. Abidemi or Monisi, please, you are next.
Thank you very much, sir. Our well-respected professor and father. Thank you for asking me to speak. Well, just to pass a few comments, we must understand the politics when it comes to international aid. I'm happy our mentor, our father is there, President Tumori, that have served at various levels, at international level. There's international politics, and that's why I support your recommendation that as Africans, we must take our destiny to our own hands. So you cannot blame the West, because people also need to protect their territory. And the truth is that we have the resources in Africa. Look at President Tumori, for example. We are serving WHO in several capacity. Make a lot of impact globally. But is there in Nigeria, not fully utilized? When I was privileged to be Vice Chancellor of Redeemer University, look at how Prof turned around that university. Now you cannot talk about molecular laboratory in the world now. Look, Redeemer University is one of the reference points. So our major issue, there are two: one, lack of political, commitments, and two, corruption. Is a major issue. I was nominated as part of a steering committee during COVID-19. I won't mention the state. In that state, people contributed money, more than 2 billion. I will make, several recommendations to that state. And at the end of the day, nothing was done. How the 2 billion disappear, only the stakeholders in the state can explain. So those are the issues. We have human resource. We have people that can, drive. Look at the guest speaker, for example. In Saudi Arabia, you are doing great work there. Originally, you're in Nigeria. But the issue is that our leaders must be willing, to support, support in the academia. And two, the issue of corruption must be addressed. Thank you, sir. And over.
Thank you very much. Mukhtar Adeza, please. Next, and then
thank you very much, Prof.
Answer the questions after this one. Others will follow. So, Mukhtar, please go ahead.
Thank you very much, Prof, and congratulations to the presenter. This was a very amazing lecture. I won't say too much, but I just wanted to look at all the issues raised in terms of recommendation. And is the presenter able to clearly define these gaps, give us an idea of how prepared Nigeria is as far as a vaccine production for emerging and re-emerging diseases are concerned, and how much more do we have to do, or who are the key responsible players in this field that should fill in this identified gaps? Thank you.
Thank you. I think we'll stop at that and let Bashir answer. Isa Musa, please, can you mute your phone, please? Now, can you please go ahead, answer those questions, and then we come from after you're finished.
Okay, sir. Thank you, everyone, for those
Bashir, please go ahead.
Okay, sir. The first questions were asked by Professor Mustafa Imam on who should be responsible for our situation. I mean, who should respond to some of our problems? Of course, ourselves. Our government should really do something about it more than anyone else. But even beyond our government, infectious diseases is something, like I said, that requires multi-country coordination, because infectious diseases, they don't really respect any international boundary. They can really spread to anywhere. So wherever it is occurring on Earth, it is our own responsibility as human beings all over the world to come together and act together to see that we solve that issue. But if it is not working, more than anyone else, like I said, we, the primary, victims, we must really do something about it. So if anyone refused to do it for us, when we ourselves did not do it, of course, we have no moral justification to blame those people. So I completely agree with you that we shouldn't blame anyone for our situation, even though just because we refuse to also help ourselves. But then infectious diseases, like I said, they require, multinational coordination in terms of response in order to make sure that it is well tackled. Secondly, the issue of surveillance that you raised is very important, actually. We have so many laboratories Thank God during COVID-19, I really say that NCDC and Africa CDC generally, they really did well during COVID-19 because they were able to activate so many laboratories. If you look, by the time COVID arrived, for instance, in Nigeria, only very few laboratories had capacity for even molecular diagnosis. But thanks to NCDC, in no time, they expanded the capacity. If you look at the current molecular laboratory network in Nigeria, you find out that we have lots of laboratories. Almost every state has the capacity for this molecular diagnosis. But the issue is, after COVID, these laboratories are not being utilized. For instance, recently we had cases of dengue, suspected cases of dengue, and this was coming from Sokoto. And this Sokoto, we have a robust center. We also have good laboratories that are able to not only diagnose, but even sequence and check everything about the virus. But then everything was centralized at the national level at NCDC. So samples had to be taken from Sokoto to Abuja to be tested for a disease that could have really been easily diagnosed right in Sokoto. So what are we trying to say? It is high time for us to decentralize some of these activities to make sure that at least in every region, if not in every state, we are able to swiftly respond to some of these things, because that is the essence. We have created molecular laboratory network. It's for COVID, but far beyond COVID, we must continue to utilize those laboratories. So this is my take. My take is there's a need for decentralization. The issue of what is different in Saudi that we need to implement in Nigeria, when surprisingly, I saw quite a lot of things here. First of all, the center where we work is a very close, it has close kind of working relationship with hospitals. So we are really keeping in track. Whatever we get from hospital, we try to quickly do research and see how we can get proof of concept and try to translate it. If it, of course, passes all the hurdles. Once we have some proof of concept, then we will try to immediately swing into clinical trials. And this they are able to do because of massive funding in research. They have a lot of funding, and they also try to buy the state-of-the-art equipment. Any latest, for instance, in terms of sequencing, latest sequences, they are available. In terms of genomic studies, like human genomics, like human genetics, they buy all sorts of things. And so they do all latest, they align themselves with the latest happenings. And then they provide scientists from across the world who would work towards achieving those things. So what we should do in Nigeria is simple. We just implement what is supposed to be done. All we need to do is equip our laboratories, and then train more people, and then provide more grants for some of this investigation. Because at present now, only a few agencies are, providing grants for research. I remember when I was during our research on COVID and research on Lassa fever, none of them was sponsored by Ministry of Health. It was all sponsored by TED Fund. And then after TED Fund, nothing. So TED Fund is the major financier, and from the results we obtained from some of our preliminary findings, and the Ministry of Health also wanted to claim it. Meanwhile, they did not really sponsor some of this activity. So what we are trying to say is there's a need for us to really invest so much in research in order to make sure that these things are available. I completely agree with the comment by Abidemi that lack of political commitment and corruption are our key problem. Then finally, Mukhtar asked on how prepared is Nigeria in terms of vaccine research. Well, as far as I'm concerned, I will say that yes, we are doing little things, few things here and there, but we still have a long way to go. Like I said, if you look at even our genomic capacity, genomic surveillance capacity is still quite low. Not to talk of, the main rigorous preclinical vaccine research before we even go into clinical trials. How many clinical trial centers do we have in Nigeria? How many even laboratories are able to do vaccine research? How many people have the capacity to even do some of these researches? So what we are saying is And our situation in Nigeria requires a multilateral approach in which we need to train lots of people in this area. Whether we like it or not, these diseases will continue to emerge and re-emerge. And until we have enough personnel to take care of it, we will always sit down and end up in this kind of situation. So there's a need for us to emphasize on staff training, for us to emphasize on infrastructure, for us to emphasize on equipment and laboratory, building, to mention but a few. Thank you very much. I hope I was able to address some of your comments.
Thank you, Bashir. I'm sure, we can go on and on and on about all this. So what you have said, 1,000 people have said it before, but the actual end is, are we going to act on it? Yes. If not, come another 20 years, you will have another Bashir, a younger Bashir, giving the same presentation, and nothing else will happen. And this is something. Let me also mention one or two things. Thank you you mentioned the issue about NCDC. When I was growing up in this country, each state had its own EP division. Epidemiological studies was not centralized like we are doing it with NCDC now. Many times I've asked the NCDC people that they should move away from National Center for Disease Control to National Center for Disease. States have abandoned their duties. A P division that were working in those days. I went to a state a few years ago. The commissioner for health was asking me to beg his governor so that he can get a vehicle for their department. So if you don't have a vehicle in a P division, how are you going to investigate epidemics when it's going on in the village? I went up to that meeting, saw in the governor's office about 10 or 20 cars, line of which nobody was new cars. And when I got there, I said, Honorable Governor, you have 20 cars waiting in this place. Your P division at Avenue has no something. At the end of it all, even the commissioner was getting annoyed with me that I brought the case up and said it the way I said it. Maybe I didn't say it the right way, but the point is that these are some of the things we need to, multilateral involvement is what we all need to do this thing. Let me give you one other example. Yes, indeed, when COVID came, we said we had only 3 laboratories. And then we build up 100, 160 or something. But do you know that 6 months into the COVID, 60% of these laboratories were no longer functioning? Why? When you give the $100, 100 naira to build a lab, and you say 50 for the lab, 30 for the equipment, and 20 for staff, some states will build a lab for 20, pocket the 80. 2 days later, the lab is no longer functioning. If you go back now, go and find out all the labs that the NCDC had created in those days, how many of them are functioning? And as you said yourself, Bashir, how many are we using? You mentioned the security about dengue. How many are we using? Everything must go to the Center for Disease Control. These are areas that we need to solve our own problem ourselves and equip those people. Get all this information. But one other important point, I think we must never underrate surveillance. That is the crux of everything, because if you can't detect, you don't even know you have. If it takes you too long to know, then the thing will spread and nobody will, it becomes a big pandemic. So emphasis will be on, and we have the resources and we have the talent, connected people who have experience in surveillance, who have been there. And we've given many examples before. I say the spare tire from my president's Escalade or whatever it is called, that his car, will buy enough vehicles for some of those states to carry out this. These are the issues we need to. What is our focus? What is our priority? Where are we going? Are there any other questions? I haven't seen any other hands that are raised. I'm still checking. Any other raised? And if you want to talk, please just unmute yourself and talk. This is a free place. This is our own country. Let us all see how we can find a solution to the problem. Any more messages? If I don't have any, I will pass on to Aminu to round off for us. Is there any other person? No? Going, gone. Aminu, the Lord's back to. First of all, let me thank Bashir again. Thank you for what you are doing. Anyway, I must say more than that. Aminu, please take over.
Thank you very much, Professor Oiwale Tomori, for your time and for sharing this interesting webinar. And thanks to Dr. Bashir Muhammad Bello for an interesting talk. We thank you all, and we thank you for your support and for the membership as well, and for words of encouragement as usual. Professor Tomori has been a father, a grandfather for so long, and the inspiration is always there every day when I'm doing things personally. Honestly, he's among the people that whenever I remember them, whenever they came to my mind, I tend to do things very correctly to the best of my ability. He's one of the top in my list, and trying to be like him and always. And so we are praying for more strength, health, so that you can continue doing the good work and support that we are doing as well. We thank you so very much, sir, for your participations and for your support. Thank you very much. I want to ask Dr. Haya Abubakar Yabo. I think he has something to add, I mean, in terms of closing remark. Thank you.
Yeah, so this is just the vote of thanks on behalf of the admin. On behalf of the admin of the AfriScience Network, I would like to thank Professor Bello for his valuable time and for chairing today's webinar and his continued support to the network. We appreciate your fatherly support, sir, and we hope that you will continue to support the network. I would also like to thank immensely Dr. Bashir Bello for your wonderful presentation. This presentation could not have been made at a better time than now. We thank you very much, sir. And we hope that the recommendations shared today and the robust discussions we have will go a long way in forging a new direction for vaccine development in Africa. Thanks to all the members of the AfriScience Network that joined today, and we hope to see you in our subsequent webinars. Thank you all.